Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video will cover the group problems for Lecture Exam 2. Remember, the group problems are not turned in for points. They are here to help you study for Lecture Exam 2. All of this information has been covered in the lecture videos, the lecture slides, the lecture notes so far. What the group problems do is they put that information in a essay form, essay problem form. And so you get to see the information in a new light. You get to synthesize and combine that information, the information from those materials that we've gone over in a different light that will help you be able to analyze this information better and be able to remember this information better and be able to apply this information better. So, group problems are absolutely necessary when preparing for the lecture exam test 2. Group problems are going to take your knowledge to the next level, the level that I expect for this college level class. All right, so group problem number 1. Outline the major steps for complete oxidation of one glucose molecule. So complete oxidation of one glucose molecule, the complete breakdown of one glucose molecule. So the steps for this are going to be glycolysis. And remember what I said about studying metabolism. You want to take each one of these steps and put it in a little box. And then learn each box and then see how those boxes relate to each other. I feel like that's the best way to learn this information. If you look at it as a whole, it can be overwhelming. But if you just learn each individual piece and then see how those individual pieces fit together, it's very manageable, very doable. Organize your learning, organize your thoughts, organize this information. So we have 4 boxes. We've got the glycolysis box, the pyruvate activation box, the Krebs cycle box, and the electron transport chain box. All 4 of those are going to be the 4 steps for the complete oxidation of one glucose molecule. You should know what happens in each one of these steps. I'm not going to go over each one of them specifically in great detail in this overview of group problems right here, but you should know where each step happens. Glycolysis is going to be cytosol, activation, Krebs, electron transport chain is all going to be in the mitochondria. You should know what goes into the process, so what's the starting point, and you should know what the end products are. What goes into glycolysis, what comes out of glycolysis. You put in a 6-carbon glucose. You also spend some money to make some money. You spend 2 ATP to get the process going. You end up with you produce 4 ATP, you profit 2 ATP. You end up with 2 pyruvate molecules, each one with 3 carbons. You should know what goes in and what comes out of each one of these. Include the specific metabolic pathway, the number of ATP, NADH, FADH2, and CO2 used or produced, so that's the what goes in and what comes out, and the total number of ATP generated in each pathway. Assume that the total number of ATP produced is 40 in this case. So this last part right here, assume that the total number of ATP is 40 in this case, is a very important line. What that does is it tells us that we are going to be using the malate aspartate shuttle for this example. So the malate aspartate shuttle, we're really going to go into for group problem 2, but I'm going to give you the kind of the rundown for it right now. So the complete oxidation of glucose using the malate aspartate shuttle which takes place in liver cells and most cells of our body, not skeletal muscles, most cells of our body. So, in glycolysis, one of our inputs is an NAD+. We need an NAD+ to do glycolysis. 2 NAD+, actually. During the process of glycolysis, that NAD+ gets turned into NADH. NADH. We get 2 different NADHs. NADH is it carries a lot of high energy electrons. Those high energy electrons can be used to produce ATP in the mitochondria. They need to be inside the mitochondria in order to produce ATP from them. These NADHs produced during glycolysis are in the cytosol. They're in the cytosol. So we need to get them into the mitochondria. This line signifies the mitochondria. Into the mitochondria in order to produce ATP. Well, with the malate aspartate shuttle, which is the one that the liver uses, no energy is lost. We have NADH on the outside, NADH on the inside. In skeletal muscles, they use a different shuttle called the glycerol phosphate shuttle. And the NADH, its high energy electrons are moved onto a molecule called FADH2 inside the mitochondria. FADH2 then goes into electron transport chain and produces energy. Some energy is lost in skeletal muscle during this process. NADH is able to produce around 3 ATP. FADH2 produces 2. So that's 1 less ATP per NADH produced in glycolysis. You produce 2, so it's 2 less overall. I bring this up and I point it out because I think that it's very visual right here in this diagram to see, and it's one part of this material that students often get mixed up on. All right, so perfect. So this will take you step by step, what goes in, what goes out. Another part of this material that sometimes confuses students are the columns for these summary charts. So these summary charts are actually really great at helping you learn what goes in and what comes out of each one of these steps. I'm going to go over what the columns mean because sometimes it's a common thing that throws students off. So ATP used is the spend money to make money. It's the ATP that you put into the process in order to get the process started. In glycolysis, we use 2 ATP to get the process started. ATP used is 2. Direct ATP is ATP that is made via substrate level phosphorylation. It's ATP that's made right then and there. ATP that does not need the electron transport chain to be created. ATP made right then and there does not need electron transport chain. NADH is a high energy electron carrier. It's going to be produced throughout the process, each one of these steps. And you get ATP from NADH via the electron transport chain. It needs to be moved into the mitochondria, go to the electron transport chain, and then that's where it cashes out. That's where you get your ATP. You get 3 ATP per NADH. FADH2, another high energy electron carrier, you get 2 ATP per FADH2. The column for indirect ATP is ATP that's created via high energy electron carriers, via the electron transport chain. So NADH, we have 2 NADH created during glycolysis. We get 3 per NADH. 2 times 3 is 6. 6 indirect for glycolysis. Activation, 2 NADHs. 2 times 3 is 6. Right here. Krebs, 6 NADHs are produced, 6 times 3 is 18, plus 2 FADH2s times 2, so that's 4 ATP from FADH2s, plus the 18 here, we get 22. Total ATP is indirect ATP plus direct ATP, so 4 plus 6 is 10. 2 plus 22 is 24. Net ATP is total ATP minus the ATP you use to start the process. Nowhere carbon dioxides are lost. All right. So that's another part where students often get mixed up, often miss points on the exam. So understand what those columns mean, what those columns signify. All right, so let's move on. That's going to be all the highlights for that question. Number 2, when we break down glucose through glycolysis, we get 2 NADH molecules. The fate of those NADH molecules is dependent upon the condition of the cell and the type of cell where they are made. Please discuss the various fates of these NADH molecules and how much energy they are worth, how much ATP we get from them. So, I alluded to this question when talking about question 1. All right, so let's go back to glycolysis. Glycolysis occurs in the cytosol. Glycolysis produces 2 NADHs. Now, if you go back to my original videos on this, I tell you to learn glycolysis. Boom, glycolysis, there's your box. And then you go either left or right. You go anaerobic or aerobic respiration. Let's go anaerobic, without oxygen, respiration, and see NADH's fate. So, in glycolysis, NAD plus is required for the process to occur. NAD plus gets turned into NADH. In order for glycolysis to continue to happen, we need more NAD pluses. In anaerobic, so without oxygen, anaerobic glycolysis right here, in order to turn NADH back into NAD+, we combine the NADH with the pyruvate, turning pyruvate into lactate and the NADH back into NAD+. This allows that NAD+ to help another glycolysis cycle occur. It also helps because the lactate can be used in gluconeogenesis. It can be used to make new glucose, which can go back into glycolysis and so on to make more energy. We don't waste that product lactate because of gluconeogenesis. Going back to glycolysis. If we take the other route and we go aerobic respiration, we go down the aerobic respiration, the next question we need to ask about the fate of NADH is, what tissue are we in? What cell are we in? If we are in the liver or most other cells, we're going to use the malate aspartate shuttle. That's the shuttle that we used in our last example. That's the shuttle that we used to move those 2 NADHs into the mitochondria, and no energy was lost. We had NADH on the outside of the mitochondria, NADH on the inside of the mitochondria. NADH, NADH, no energy was lost. And then that NADH was able to go on to electron transport chain and produce free ATP. If we're in skeletal muscle, we're going to use a different shuttle to move those high energy electrons into the mitochondria. We're going to use the glycerol phosphate shuttle. The glycerol phosphate shuttle is going to lose energy. That NADH on the outside of the mitochondria is going to become FADH2 on the inside of the mitochondria, which only produces 2 ATP per molecule. So some energy is going to be lost. 2 ATP is going to be lost. I want to go back, and I actually just thought of another thing that students often get mixed up on in the exam. So this example here, and all of the numbers in the summary chart, are following the story of one glucose molecule. What would the numbers be if I did it for 2 glucose molecules? All the numbers would be doubled, right? Well, what if, so glucose gets turned into 2 pyruvates. What if I talked about the activation of 1 pyruvate? How many carbon dioxides are lost when you activate 1 pyruvate? If you look at the summary chart numbers, it's talking about 2 pyruvates. So it would be half of what the summary chart is. When you activate 1 pyruvate, you lose 1 carbon dioxide. What about 1 turn of the citric acid cycle or Krebs cycle? In this diagram right here, and in the summary numbers, the summary chart numbers, we do 2 turns of the citric acid cycle or Krebs cycle because 1 glucose allows for 2 turns, 1 per pyruvate that gets activated. So on the test, I might ask a question like, for every 1 turn of the citric acid cycle, what's produced? So be ready for those. I want you to understand this at that level. All right, moving on to 3. Discuss Cannon's 4 postulates in regards to homeostasis. Please explain each postulate and provide examples where appropriate. Students tend to do pretty well with Cannon's postulates as long as they can remember them. As long as you can remember them, students tend to do pretty well. So I recommend that you make flashcards. I recommend that you practice recalling Cannon's 4 postulates. Most of the time, I find that if I tell the students what the postulates are, they can tell me all about them. But if I ask them what the postulates are, they might get 2 or 3, but that 4th one might be a little tough. So practice before the exam. First postulate, the autonomic nervous system plays an important role in this process. So we've talked about the autonomic nervous system a lot by this point, from heart rate to sweat to, it just goes on and on. So be ready for some examples there. There's always a level of tonic activity. Now, whether it's the nervous system or the humoral system, hormones, there's always some level of each hormone in our body. We never completely stop making them. When I'm scared and I'm running from a bear and my sympathetic nervous system's going crazy, my parasympathetic nervous system never completely shuts off. It just decreases the frequency at which it sends signals. It decreases its stimulation. There are antagonistic controls. We've talked a lot about this, from insulin and glucagon helping to control blood sugar. One raises blood sugar, one lowers blood glucose levels. To the heart rate, and the parasympathetic and sympathetic nervous systems working antagonistically. Know your examples. It is the receptor that gives the effect. It is the receptor that gives the effect, as the same chemical could have different effects on different tissues. So this one, you're really going to want to look at the end of the nervous system lectures. At the end of the nervous system lecture, we talk about the efferent nervous system. And we really see some good examples about how the same neurotransmitter can cause different effects on different tissues based on the presence of those receptors. Now, there is a group problem on the efferent nervous system, so we'll get to those neurotransmitters and receptors. I expect you to know what neurotransmitters are where in the efferent nervous system. That actually has a lot of implication in healthcare and medicine in general, so definitely know that. Also, know the receptors, because once again, huge impact in healthcare. Knowing that will have a positive impact on the patients that will be in front of you in the future. All right. So moving on. Explain the three major ways the body communicates. Explain the specific mechanisms, both neural and humoral, within each method and cite each example. So students tend to do pretty well on this one also. There's not really any major spots where students tend to miss things on this one. So direct cytoplasmic transfer, just what the name implies. There is a tunnel, a direct connection, a tube connecting two cells, allowing for ions to pass or various substances to pass. So chemicals such as metabolites, metabolic substrates and products, as well as salts, can pass directly from one cell to another via gap junctions. Electrical, since one form of electricity is really just the movement of ions across the cell membrane directly, ions such as sodium can pass an electrical event directly from one cell to an adjacent cell. So when we study the cardiac muscle, the heart, in our next section, we're going to see how these are really important for transferring, for coordinating contraction within our heart. Local chemicals. So typically endocrines, so local chemicals can be within one tissue. They're not going to be far off tissues, can be with far off tissues. It's chemicals communicating, coordinating function with their neighbors. So autocrines, chemical release from a cell that acts back on itself, such as histamine. Paracrines, chemicals released from a cell that act on neighboring cells via diffusion. Histamine could be an autocrine, it could be a paracrine, depending on where it binds. Neurotransmitters. So neurotransmitters cross a synapse. Synapses are incredibly, incredibly small. Local communication. Even graded potentials, and those IPSPs and EPSPs are considered local communication, local chemicals. Long distance communication, this is going to be what most people think about when they think about communication within our bodies. It's going to be hormones, a chemical released into our bloodstream which sends a signal to a far away part or a different part of our body, and neural, which is going to be neurons sending action potentials down their axons, or that electrical signaling. Explain what is happening in a normal nerve cell. Make sure to touch on how the resting membrane potential is established, what is the equilibrium potential of each ion, how equilibrium potentials are established, and how they are maintained. Also cover threshold potential, depolarization, repolarization, hyperpolarization, paying close attention to their properties based on the relative permeability of ions. Draw a graph that shows the typical voltage for resting membrane potential, threshold potential, peak action potential, and hyperpolarization. Explain what is happening at each point with respect to the major ion channels responsible for these voltages. So, this question right here reminds me of my advice when I talked about the learning cell metabolism. When you look at this whole problem, feels a little overwhelming. Take it piece by piece. So, how is the resting membrane potential established? The resting membrane potential is established because of the sodium-potassium pump. The sodium-potassium pump creates an electrochemical disequilibrium across the nerve cell membrane. It's pumping sodiums out, it's pumping potassiums in. Then there's also leak channels. These channels allow sodium to come back into the cell at a certain rate and potassium to leave the cell at a certain rate. Potassium is able to leak out faster than sodium is able to leak in. This sets up a situation where there's more negative charges inside the cell, more positive charges outside the cell. More potassium inside the cell, more sodium outside the cell. The resting membrane potential is going to be -70 for a typical neuron. That resting membrane potential is going to change when we change the permeability of the neuron to each of these ions. If we allow sodium to rush back into the cell at a different rate, it's going to change that membrane potential. If we change the rate at which potassium is able to move across the cell membrane, we're going to change the membrane potential also. So when we open up additional potassium channels, potassium rushes out faster. This is going to hyperpolarize the cell. It's going to cause an IPSP. It's going to make the cell more negative because we're removing positive charges from the cell. If we open up an ion channel that allows sodium to rush in, we're adding positive charges to the neuron. My power just went out, so sorry if you can't see me in this video. I'm going to pause this.